It's a real honor to speak to you today. I'm going to talk to you today about the 100,000 Genomes Project and how we've used it to transform healthcare in the NHS. So these are my disclosures. I haven't received any money from anybody really, other than my salary. So, and I work for Genomics England, seconded from Queen Mary University of London. So that's a public company, so it's owned by the UK taxpayer. The origin of the 100,000 Genomes Project started as a legacy program from the London 2012 Olympics. And in essence, what that means is that David Cameron, the then Prime Minister, gathered together scientists, health ministers from around the world during the Olympics, and they discussed what would be major advances that could be part of the Olympic legacy. So this led to the 100,000 Genomes Project, and we've had successive support from government. The Genomics England was launched on the 5th of July 2013, the 65th anniversary of our National Health Service, which is free at point of care. And you can see here various government ministers and officials who supported the programme. And I show you this slide. We don't normally show pictures of our politicians in Britain because we're not entirely happy with them a lot of the time. But I'm showing you these because these people are stuck by bringing health service genomics live in the health system for now the best part of almost 8 years. So the 100,000 genomes, we are reading whole genomes, not parts of genomes. So we're reading as much as you and I can of the 3.3 billion letters that makes you the individual you are, but also can sadly carry susceptibility to disease. Expert groups decided that these should be focused on rare disease, cancer, and infection. And we've reached over 97,000 patients and their family members from across the National Health Service. We assembled initially 21 petabytes of data. Today, we're at about 40 petabytes. And to do this, we had to create the infrastructure in the health system. So we arranged for the National Health Service to commission 13 genomic medicine centres of excellence spread equitably across England, and that reached 98 hospitals. At the peak of the programme, 5,000 frontline health system staff worked on the programme across a range of disciplines. Because you and I today don't absolutely know how to use all of this data in healthcare, we openly advertised in Nature on 2 occasions and created a coalition of global research intellects with 3,000 volunteers from 33 countries. So on this slide, what you're seeing is a map of the UK. And this project was initially focused on England. And you can see in the red dots, the 13 genomic medicine centres. And you also see that Scotland, Northern Ireland and Wales, I got them involved in the project. So in essence, this reached the whole UK and had regional equity of access to anybody with appropriate disorders. Patient consent was obtained in the genomic medicine centres. They then submitted samples and clinical data alongside collection of longitudinal life course health data from electronic records. And we then assembled their samples in a biorepository, which is the UK Biocentre, and we built a sequencing centre at the Sanger run by Illumina on our behalf. And we openly competed to have the sequencing done by the company. And this has been an extremely good partnership where we've had the lowest price in the world, access to the latest technology, the latest chemistry, and even better, Illumina have done the work for us. That's worked very well. We also created a large data center, and this data center is where researchers from around the world can come and work through your desktop as a virtual desktop on all the data, and you have free access to that. We also created an industry consortium to work on the data and to do research with alongside academics that would drive up the value of this data for clinical care. Finally, a report is returned to the clinician in the front line, and they own the relationship with the patient and decide whether the diagnosis is returned or not. So on this slide, what you can see is a genome and its journey. So we sequence the genome in rare disease for the germline at about 30x. We achieved a median of 36 to 40x. And for the somatic genome, in this case, the cancer, we sequenced at 75x. Because you want even coverage, you have to over-sequence, so you end up with about 82 to 100x, depending on the cellularity of the tumor. Now, you see that the patient and family were consented, and their DNA molecule, which if you click on your screen may flow around the path, The DNA goes into the system and the genome is sequenced. We then quality assure the genome. We then annotate the variant files for potentially causative mutations. We had some annotation companies supporting us during the program, and eventually we tier the variants according to their likely pathogenicity and then whether they could be plausible but are not unknown variant for the disease. We then prepare a report and return it to the clinic for assessment. Simultaneously, we place the genome and the clinical data in the research environment available for patients. We're now going to focus on rare disease. So if we go to the next slide. In rare disease, that affects about 6% of Western societies, about 1,200 disorders with unmet need. We standardized eligibility in phenotyping, and we used the human phenotyping ontology, which was in fact developed originally at your university by Peter Robinson. And that allows us to standardize data collection. We used automated analytics and NHS confirmed gene panels and closed cases. On the right-hand side, there should be a picture of a family, but unfortunately the animation isn't working, so I'll talk you through this. So this family of 3 people, there was a child who was enrolled about their 4th birthday, and they had intellectual disability and uncontrolled seizures, so an epileptic encephalopathy, and no cause had been found with usual care testing. What we were able to do is to show that we could actually develop a whole genome for that child and then look through the genome. So if you imagine there's 3.3 billion letters here, we found 6.4 million variants, of which 677,000 are rare, 2,826 changed a protein, 67 were different from mum and dad, and 1 was a clear-cut cause of this child's condition. So we do that every day in about 10 hours. In this child's case, it revealed that there was a glucose transporter 1 mutation, which meant the child could not transfer glucose from the bloodstream into the brain. And this explained the seizures, because every time the brain sugar levels dropped down, she had fits. So this paved the way for a ketogenic diet, a high-fat diet, and as a result, she is much better with fewer seizures and has shown some improvement in her intellectual disability. At the bottom, you can see the various different disorders we've enrolled against. So on this slide, you will be able to see a range of diagnoses. I'm going to take you through a few of these just to show you the power of whole genomes. So the first case is a really sad one. This was a 4-month-old who was born, went straight to the neonatal intensive care, and sadly died with no diagnosis. Immune testing was negative. He was enrolled by his parents in the project after he died with DNA that had been stored. And they really wanted an answer. They were closely related, first cousins. And then the mother unexpectedly became pregnant. She said, look, I don't want to know. Don't tell me what's wrong, because I'm going ahead with this pregnancy. So at month 8, she became pretty anxious and asked us to have a look at the genomes. And we found a pathogenic transcobalamin 2 mutation, which allowed us to have a diagnosis for that child. When the brother was born, this meant that the National Health Service could test him within 1 week. They diagnosed that sadly he was also affected. Now, what transcobalamin 2 does is it transfers vitamin B12 from your bloodstream inside cells. And there were case reports, which we reported on, that had shown that if you gave very high doses of vitamin B12, you might be able to avoid some of the adverse effects of this disorder. And that's what happened. And this child has remained ostensibly well since, so has had nothing like the course of his older brother. So this is an example of a genome paving the way, shining a light on a potential therapy. The next is a 10-year-old girl admitted to ITU with life-threatening chickenpox and prior unusual severe infections. This child had cost the NHS £356,000 with multiple ITU admissions. Immune testing and other genomic testing had shown nothing. We found mutations in fact, CTSP1, not CTP1. That's a CTP synthase gene that affects B and T lymphocyte function and paves the way for infection with both capsulated bacterial organisms and viruses too. So this child had the opportunity then to have a curative bone marrow transplant at the cost of £70,000. Imagine if we could have found this shortly after birth or on the first presentation, we could have saved a lot of money with ITU admissions, and her siblings were tested and not at risk. The final case is a child where the diagnosis reverses the clinical suspected diagnosis. So this is a child with unexplained anemia, developmental delay, short stature and constipation, who'd been given a diagnosis of Diamond-Blackfan anemia. This is quite a severe diagnosis which may have a limited lifespan and cancer risk. But because this child had developmental delay, we deployed the panel that looks at intellectual disability, and that showed that he had a thyroid hormone receptor alpha mutation, and he's now receiving high-dose thyroid hormone replacement. You can see a video of this child on our website, and you can see that he starts being unable to walk normally, and by the end of the film he's dancing. He can also speak much better, and he's shown some improvement in his intellectual developmental delay. So this is an example of a genome reprofiling a diagnosis. So I'm not going to pretend to you this happens for everyone, but these give you some examples. In the next slide, I'm showing you the words of a mother of this child in the picture on the right-hand side. We made a diagnosis in this 5-year-old of GAMP deficiency. This is a treatable metabolic disorder. Her mother was suspecting that this was a severe degenerative neurological disorder, which it appeared to be. She had intractable epilepsy. She was locked in, unable to communicate. But after 6 months of simple dietary supplements of creatine, ornithine, and arginine, this is a new child, and she now can communicate, and she's free of epilepsy. She'll never be entirely normal, but the last paragraph is the mother's words. And if you see, as I'll show you in a moment, the diagnostic odyssey these people go through, you will understand why this means so much, even though her child may not be cured. So this is PanLab, which we published in Nature Genetics. So what we did here was we wanted to source as much of the knowledge we could worldwide. So we openly called for people to volunteer genes and evidence for variants that should be included in an initial search through the genome for efficiency. And you can see here in blue the countries in the world that have contributed, Germany included, to development of PanLab. And it's now been used by over 570,000 different requests. There may not be different people. And what this does is it's allowed us to create the annotated panels with evidence from all of you. We've been able to crowdsource that knowledge and then make it available back to you to use in your genomic analysis as a service to humanity. So if we go to the next slide. So we use this in our genome analysis every day. And what we've also found, and this is from the first 2,183 families in the program, that we can achieve a diagnosis in about 25% of families, in some instances, in the pilot phase, and that if you have increased family members, mum and a child duos, or both parents and a child trios, you get increased line of sight on a diagnosis. Also, if you look at the right-hand side, you'll see that the diagnosis is up at 36% for likely single gene disorders, and likely complex disease etiologies have a lower diagnostic yield, as you might expect, at about 11%. On the right, the pie chart shows a appearance of what you can see if you just were to look at panels in blue, and then the yellow, green, and orange, all the things you get from the genome which you won't get from the panel, giving you an idea of how you augment genomes. And we now have many diagnoses that would not have been detected with exomes, so the whole genome is a superior exome. So this is the diagnostic yield by trait or system, and you can see the threshold line for 25%. You can see hearing and ear, metabolic disorders, intellectual disability, neurology, ophthalmology, all achieve greater than 25% diagnostic yield. And this correlates with the conditions that we brought live in the NHS for whole genome sequencing. Here is something I did not think we would achieve. So this is looking at simple tandem repeats. On the left of your picture, there is the normal repeats in the blue histogram. So what we've done is we've used software algorithms to measure the repeats across all the genomes we have and define what the normal distribution of that repeat would be in the population. The red dotted line, on vertical line, shows you where the repeat begins to expand. And in red shaded triangles are people with diseases that have simple tandem repeat-based disorders and repeat expansions. So you can see here different repeat expansions. We've now validated these for 13 commonly called STR repeats from a short read genome, which I did not expect us to be able to do. So here you can see a taxin 1, a taxin 2, a taxin 7, And at the bottom, you can see Huntington's disease. So one of the cases with the shaded red triangle on the right-hand side between 90 and 100 repeat size expansion, that is a 10-year-old child who has been enrolled with hereditary ataxia but was never suspected to have Huntington's disease. So if we detect that child earlier in his course, maybe they could have had the antisense oligonucleotide and possibly have had some reduced disability or maybe delayed progression. So this turned out to be a transmission from the father, and the repeat has massively expanded from the father. The father is over near 40 in the open red triangles, but the son is over at 100 and massively repeat expanded. So this is known in Huntington's disease, but it paves the way potentially, possibly for treatment. But these are early days in knowing whether that will be effective. I wanted to show you what the diagnostic uplift is over standard of care that's used across the world. So we looked at some cases from the pilot where we could obtain testing or no testing data. In other words, we're confident they didn't have a test or confident they did. About a third uplift in your diagnostic yield, whether you have a test or not, from prior testing. So, and you can see the different uplift in diagnosis by a whole genome, including on the right-hand side, only small numbers of cases, but 12 people who had a WES whole exome, there's still a substantial uplift in diagnosis. So whatever test you use, a whole genome is better at diagnosing the disorder. So this is a report that they get. They get the pedigree back. They can make changes in the pedigree. They get a report describing the pathogenic mutations tiered to known pathogenicity, likely pathogenicity, or possibly pathogenic, but not a gene we've seen before, for further annotation and validation. And they can look at the raw data, including the BAM files in our system, and also at the variant call files. The data from our pilot had immediate clinical utility in 24% of cases. For 3 probans, it changed medicine. For 20, it led to additional surveillance. 30, and it led to a clinical trial. 52 had reproductive choices informed, and 32 had a dietary change or vitamins. In addition, from cohort burden testing, we discovered 500 genome-wide significant signals for which we need further functional annotation, and I'll come back to that towards the end. So this is the diagnostic odyssey that children in this pilot of 2,000 cases underwent between 2003 and onward. And we used 2003 because we wanted to focus on the period where we had electronic health records and could follow the children comprehensively. So families spent 6 years, a median of 75 months, attending 68 hospital appointments without any diagnosis whatsoever. Unaffected relatives from the same families, by comparison, attended 18 appointments over 120 months from birth. After diagnosis, what you see is a much more focused use of the appointments targeted to investigations or potential therapies that might help the child. So in total, affected participants used 187,000 episodes in direct healthcare, costing £87 million, and that's about £15,000 per participant. Now, I emphasize to you that we are now in the era of the less than $800 genome. If that's so, then £15,000 not to get a diagnosis doesn't seem very good value. By comparison, their unaffected relatives had 53,000 episodes at a cost of 21 million. That's £4,000 per participant. This doesn't include visits to general practice or disease treatment costs. So we could go to the next slide. So for all 2,183 families, they consumed 230,000 hospital episodes at a cost of £106 million. And in many cases, in all cases, that was not to get a diagnosis. And so we now be able to bring earlier diagnosis in the NHS through this modality. If I could show you the final slide on rare disease, and I'll move to cancer. So this is something called clinical variant art, which is available to the NHS and has become available to researchers. And that shows you across 36,000 families the diagnostic yield after the NHS has decided to return the result and confirmed that they agree with the diagnosis or not, that's their choice, is 20% at the moment. That's not good enough, and I'm about to conduct a major reanalysis of the whole genomes, indeed in partnership with the Berlin Institute for Health, involving multi-omic analysis, and I'll come back to that near the end. I'm going to focus now on cancer and making whole genomes work. Now, cancer is a disease of disordered genomes. Here we are sequencing the tumor and comparing it to the germline genome and subtracting it to work out what the drivers to the tumor are and what variants might pave the way for stratified healthcare. So we faced an immediate problem. We couldn't make formalin fixation work and give a consistent quality genome. So we re-engineered 400 molecular pathology pathways to fresh tissue supply for biopsies and for tumor resections. Alternative fixatives such as PAXgene give good equivalent results almost to fresh tissue. And we also have a method where you drop a biopsy into saline and just shake it like this for a few minutes, and the cancer cells preferentially fall off the biopsy because they're not as adherent, and that improves DNA yield from a small biopsy. If we go to the next slide. To emphasize to you the impact of formalin, I'm going to show you the same genome twice. So on the left-hand side, you're seeing a fresh tissue genome arranged in what's called a circus plot. And on the right, you're seeing the same person's genome after exposure to formalin. So left is fresh tissue, right is formalin. What you can see on the left is there's relatively fewer structural connections. These are the lines between different parts of different chromosomes across the middle of the central of the circle. On the right, it looks like a child has got a crayon and scribbled on the genome. It's a mess. And these are spurious structural variants created by formalin cross-linking. You also notice that the genome on the right is more uneven in its coverage. And if you go down to the table at the bottom and go to the right-hand side, you can see that the SNVs, there's 698,000 in the formalin genome compared to 10,000 of interest in the coding region of the fresh tissue genome. The same for indels. So this is not a good quality product, and so all the genomes we have now are fresh tissues. So this shows you the cancer disposition across 17,500 people. Obviously, common cancers dominate, but we've got reasonably repeatable numbers. This is the largest freely available to researchers cancer resource in the world. And you can also see in the next slide the staging of the cancers across the 13 different regions in England. So we've got stage 1, 2, 3, and 4 in different proportions, and that allows us to make observations at all stages of cancer. So I just wanted to show you our pipeline. On the left, we've got germline. On the right, we've got tumor. We use dragon alignment from Illumina, and this generates variant call files for different types of variants. We combine small variants and look at them and annotate them and variant prioritize them for the germline. Then we analyze the somatic genome by a similar methodology, and then we filter the variants from the germline to give us the drivers and those that might be relevant for therapy. So you see different cancers across the bottom here, and up the side you've got tumor mutational burden as the number of mutations in the coding region. And the dotted line, the box with the dotted line around it, is showing you where we cross to high tumor mutational burden, which may profile the patient for immunotherapies in the clinic. So you can see that endometrial cancer, colon cancer, bladder cancer, lung cancer, and malignant melanoma, and several others also have some patients with high tumor mutational burdens, and this may signal the need for immunotherapy. The two at the top of the colorectal cancer are overlapping, and these are POL-E mutations, which are known to be a source of instability in the genome. So this shows you mutational burden. If we go to the next slide, I'm now going to show you that we've annotated the genomes with rich clinical data from primary care, hospital episodes, and outcomes. And in each row on this slide is a person, and in the blue dots you're seeing non-cancer episodes, and in the red dots you're seeing cancer episodes. So you can see that some people had only one cancer episode and maybe cured. Others have had multiple cancer episodes, and some are not free. This allows us to look at real-world applications of therapies and compare the outcomes of patients to the value of that therapy in the clinic against the genome. So we can follow these people in a rich longitudinal data set. So I'll go to the next slide, please. We've also used this data to use it in clinical trials. what you can, well, actually, before we do clinical trials, we'll show you a case. So this is a family in the program. The lady in the circle shaded in green had breast cancer, but no family history whatsoever. So she presented with breast cancer and had a whole genome. The whole genome found that she had BRCA2 in her tumor, but also in the germline. So she got the opportunity to be in the Olympia trial with olaparib. Her daughter decided to have predictive testing and is a BRCA2 carrier with a pathogenic mutation. And she's now entered intensive breast screening and is considering other options. And the males in the family with no family history of breast cancer or prostate cancer could consider BRCA2 testing for prostate cancer. So this shows you the family-wide benefit of some of this data. I'll show you another case in the next slide. So this next case is a 42-year-old with colorectal cancer. And on the right-hand side, you can see a CT across his abdomen. And on the left, you can see the liver, and you can see a very dark gray region in the liver. That's a colorectal cancer deposit. It's an enormous deposit. And what we can do with that is to biopsy it, as we did. In usual care testing, no variants of clinical significance for treatment selection were shown. And so he had a whole genome because his lesion expanded and he was feeling more unwell. And this revealed that he had previously had Lynch syndrome, mismatch repair, and had the MSH6 mutation. And this means he's been profiled for immunotherapy trials. So this is a hereditary predisposition in his germline to cancer. He's just a late presenter. What if we could have found that at the outset? So here on this next slide, and thank you for expanding the screen, I'm showing you what happened in the NHS with this data. So this is the West Midlands Genomic Tumor Board and what they did with the data. On the left, you can see there's 3,244 patients. 181 patients were re-referred back to the tumor board in the central column of boxes. Of those, 477 had a tumor board action. So that's 61% of the ones that were referred back. And you can see on the right-hand side the different uses of that. So DPYD predicts toxicity with 5-fluorouracil and capecitabine. 11% got a benefit from that. 4%, 21, got a licensed therapy they wouldn't have received. 204 got in the trial. 11 got an unlicensed therapy. 43 had a high tumor mutational burden report, paving the way for potential immunotherapy, and 69 got referred to clinical genetics. So that's 25% of the patients that we looked at who could receive something potentially that might be actionable. There were a proportion of those that didn't have a benefit, but remember, we banked the genome. So if the large section in the middle, which were early resections, the 46%, then recur, the genome is there to potentially help profile the tumor. And we could resequence the genome to work out how the tumor has evolved in metastatic deposits. So we've also used the data to spot enrollment in clinical trials. So this is the Focus 4 trial, a trial of colorectal cancer when it's metastatic. So the blue dots are showing you patients in different parts of the country that have colorectal cancer and have the mutations that the trial is using to select therapy. There are some red dots here which will be difficult to see because it's not full screen, but it doesn't matter. Those red dots are people who are clearly eligible already. So we can return to the clinician a report that says, by the way, this person could be eligible for a trial. And for every single report returned, we search clinicaltrials.gov to see if there is a clinical trial the patient could be in, and then return it to the Genomic Tumor Board. So I'm going to focus a little bit on infection. So we also sequenced 3,000 TB strains, and this has brought live a whole genome sequencing for TB as routine care in the NHS. And you can diagnose within a few days the TB organism. You can profile within a further 24 hours the organism's resistance. So you can tailor anti-TB therapy now to the likely success of the therapies for treatment of the patient. So we also did pharmacogenomics and used the Clinical Pharmacogenetics International Consortium consensus guidelines and looked at the first 60,000 whole genomes. It turns out 100% of us possess a CPIC actionable gene-drug pair. And across 60,000 people, there are a median of 4 gene-drug pairs. And you can see here different things that you can detect, some of which are obviously common variants. But what if we could dissect the architecture of pharmacogenomic response and return a report on every whole genome, and then create an app which you carry around on your smartphone which you can dial in the drugs as your clinician wants to prescribe them and get a profile saying on a traffic light system, you should have this because you're green for pharmacogenomics, you probably should have a dose change, you're orange, or you shouldn't have this at all because it will be harmful. So that could be a major advance, and we're thinking of bringing that live in the NHS. Okay, so we're going to move to the Clinical Interpretation Partnership. This is the research coalition of intellects. There's 3,580 people from 33 countries who volunteered to be in this. That's 430 academic institutions. And you can see Germany there in the heart of Europe. And £50 million have been won in grants. We also have 130 companies we work with. But I'm not happy with this yet, because the grey is the sections of the world where they're not engaged. I'm not really bothered about the Arctic or Greenland, because there's not many people there. But I am bothered about South America, Africa, and Southeast Asia, because this suggests that we haven't yet reached those parts. And this data is accessible. Now I'll show you in the next slide the data that you can have access to. So you have access to 3.8 billion clinical data points of electronic health data gathered on 111,000 genomes from 90,000 participants across all sorts of primary care, hospital episodes, patient-reported outcome measures, various registries, including cancer registries, so you know what chemotherapy they've had. And we've recently got ITU registries across the UK included as well. So it's a rich data asset, and we'll longitudinally follow these people for their lifetime. So how have we used this to transform healthcare? If we can go to the next slide. So this is the anatomy of the National Genomic Medicine Service. We use the data I've shown you to transform that. And so in the middle, you see the structure. We now have Genomic Medicine Services Alliance, which is our regional centers interacting directly with patients there where the clinicians are, and a network of 7 laboratory hubs that do all testing other than whole genomes. We will concentrate all genomic testing in England in a single national genomics research library, and everyone will be offered consent for research. We will also continue to provide the whole genome sequence, the clinical interpretation of that, and manage industry and academic partnerships with people like you internationally. To do this, we created a national test directory. It's not a guideline or a catalogue, it's what you get reimbursed for. So we reprofiled 300,000 tests to do this, upgraded 25% to new technologies, and 500,000 whole genomes are available for clinicians to order in the NHS over the next 5 years. And we started sequencing on the 2nd of November 2020. Everybody will be offered consent for research, a very simple consent form. We have 2 sides of A4. First side is patient choice for a test, second side is consent. And that allows longitudinal life course follow-up for you and I to work on any exposure they get or disorder, recall for research, and access of the data from a secure data center in the UK to international researchers and industry. So here you see the distribution of tests as commissioned in the NHS for NHS service. And obviously, whole genomes are not necessarily got the evidence base to be ubiquitous. There's a mixed economy, and I suspect the mixed economy will maintain and sustain for some time. In cancer, we're using large panels mostly, but we're trying to actually expand whole genomes with new evidence generation. So we've also got a national genomic informatics system, which is how you choose your test. So you put in the condition, it tells you what test you should choose. This is to democratize access, and it's expanded across healthcare professionals more generally. I just wanted to share with you briefly some work we published before Christmas last year in Nature on COVID. So we have a program, we're sequencing 20,000 severe COVID patients who've been in ITU and comparing them to 15,000 mild and asymptomatic participants in a study where we're surveying them every 3 weeks to measure their antibody response and the antigen, the virus. So you can see that 30% of this study, which was genotyping only, 2,224 ITU cases, 30% were women, 25% were from ethnic minorities. I was very pleased by this, and 22% had sadly died. So if we go to the next slide, next slide is a Manhattan plot of chromosomes arranged 1 to X, and red serrated line is genome-wide significance. And you can see we have 7 genome-wide significant loci for severe COVID response. 3 of those are potential therapies. Interferon has been trialed and not succeeded in severe infection. Tyrosine kinase inhibitors have some evidence for them, but we've got another limb of a new trial starting with baricitinib. And chemokine receptors 2 on chromosome 3 has a biologic tested in rheumatoid arthritis. We did Mendelian randomization, and interferon 2 and tyrosine kinase have a causal link to severity. We put public and patients at the heart of the program, so we go to the next slide. So we have 30 strong participant panel who is spread throughout our organization, watching what we do, informing our strategy. And they were asked, what are the benefits of this in terms of an NHS social contract? That is the contract between the NHS and the public in the UK in a free point of care system. And they highlighted that patients should have reciprocity, altruism, and solidarity with the health system in delivering a genomic medicine service. But they also said there were some red lines. They were not wild about genetic manipulation, i.e. CRISPR-Cas9. They did not want a surveillance society or commercial use for healthcare if it was simply for marketing. They understood the need for commercial entities to work on the data and fully supported it if it was for development of diagnostics and therapies. We also have surveyed 1,886 adults between 16 and 75 in June 2019 to look at their appetite for genomic testing more generally. And 72% of respondents said they would like their pharmacogenomic profile. 71%, surprisingly less, were interested in their current medical conditions. And you can see here that there is a strong community appetite for this information now, and a building knowledge base when we test it of genomics, though obviously it's still at a formative phase. So we've got a new program that we're going to be doing over the next 5 years, and that's based on a national strategy which you can read on the web called Genome UK. That is our government's 10-year strategy for the incorporation of genomics in direct healthcare, and it's available free on the web. We're going to look at diversity. There's not enough people from ethnic minority groups involved in genomic research yet. So we're going to focus on that and pharmacogenomics. We'll have another cancer program. We're also going to do work on newborns because we've done an extensive piece of work on fully penetrant conditions. And it turns out there are 1 in 190 live births in England, that's 9 children born every day, who have a fully penetrant disease predisposition where there is an intervention that's known already, maybe dietary, maybe vitamins, might be a treatment that could potentially reduce disability. And we're hoping that by testing this alongside routine care with the heel prick tests, which only measures 9 things in Britain, that we will be able to usher in an era of genomic prevention. That has to be research with ethical and societal debate, and we'll continue to work on rare disease. So we'll also work with any country, and I'm delighted to say we're about to work with Claudia Langenberg and Marcus Rauser on a combination of proteomics, low abundance, high abundance proteins, where you at Berlin Institute for Health will be directly involved, and I'll be combining that with transcriptomics and the whole genome. So, ladies and gentlemen, the National Health Service has a genomic medicine service that will in the future provide equitable care for actually it's 57 million people now in England, and we'll spread it across Scotland, Northern Ireland, Wales. It's standardized with a standardized single national genomic consent for NHS care and research, delivering to an approved national test directory and building a single knowledge base for the UK. And that will be de-identified where they give consent for academic use and industry. And we also have a cohort called Our Future Health of 5 million people. That's the successor to Biobank UK. And the future, I believe, and that's why I'm here this morning with you, and I apologize for my slides, is a global coalition of intellects driving this into healthcare. And I'd just like to show you one more slide. And that is, these are our participants, or some of them, and they want you to see them because they're hoping you will turn up in our data center and get a diagnosis for many of these who've yet to get an answer. I should like to thank my funders on the next slide, finally, and also all the people who made this possible. Please do get in touch if you want to know more. Thank you.
Mark, thank you very much. That was a wonderful presentation. You were actually very clear. We could hear you very well. And also the presentation came through very visible, I think, on the screen. So no worries about that part. And it's certainly our technical issues rather than yours. So don't worry about that. Thank you very much. I just wanted to remind the participants that now through the chat you could enter a few more questions, although we already have received quite a few. So So while that is happening, I might just take the liberty to start asking first a couple of questions. Mark, with the understanding of what you are doing technically, how important are long reads in the long run?
Yeah, I'm sorry, Christoph, I was trying to speed up a bit at the end, so I didn't concentrate on that. But let me share with you, we are working on long reads. So we're doing work in rare disease. We have a partnership with France Genomics and Jean-Francois Deluge and Frank Leth-Monnier, and we're working with them on long read technologies with Oxford Nanopore. We envisage working now, it's more user-friendly and more affordable with Pacific Biosciences, but we've not done any work to date. We definitely see in long read genomes increased structural variant calling and a better quality genome through some of the more challenging regions. So I'll give you a pharmacogenetic example. Cytochrome P450 type 2D6 has a pseudogene issue where it gets multiple copies of the gene. This can be really challenging for the short read technologies, and although there are automated algorithms that work for this, we have found a long read genome may be better there. We're still working on this in cancer. We will have a major cancer program with Oxford Nanopore over the next year, 18 months. And this will establish the relevance of long reads versus short reads, or long reads and short reads combined, because that may well be the future for some time. But bear in mind at the moment, I didn't say this, but all our pipelines, all the way from sequencing, sample provision, all the way through to result provision, are ISO accredited for clinical care. Today, you couldn't do that with the long read technologies. So we have short read technologies for some time to come, and they do a good job. You know, Illumina put huge effort in. You know, I think the partnerships work really well.
Yes, and along the same lines, how then do you see the role of information coming from different analytes, say RNA, in proteomics? So how much is this going to be then a combined analyte analysis as opposed to just a genomic program?
I think quite often, Christoph. So let me give you an example. We've done, as part of our work, we've incorporated a software suite called Splice AI, which I didn't have time to describe. And out of about 37 patients where Splice AI, what it does is it looks at every splice junction in your and my genome, Christoph, and then it compares it with everybody else's data in the entire resource. And so what you get is, instead of 38,000 families to look through every splice junction, which is impossible manually, you get a readout of the ones that are different. And then you can focus in on bioinformatics. But we're also doing transcriptomics on those. And out of 37, we had about 8 that didn't produce good read-throughs of the splice junction region on RNA-seq. But the rest, we had 50% uplift in diagnostic from the Splice AI. These obviously were cases that were chosen because we had a reason to suspect they might have a splice junction issue, and the Splice AI had already highlighted them. But I can see that the future is multi-omic, and that's why we'll be doing an experiment with your institute.
Okay, so then a very interesting question that came in from the perspective of patients. There's a couple of questions. First, the question from Sabine Klaassen. At what point in the analysis is family data then included? So how can you access family data going beyond the range of the actual participant?
So the family data, clinical data, is collected on the parents, but obviously they don't have clinical phenotypes relevant to the disease. So every parent gets longitudinal life course follow-up, as well as the individual participant. The pedigree is drawn at the outset, and it's validated with the clinician before we go into analysis. So the HPO terms are used to help select the panels that we focus on initially, but we do range genome-wide now. So for example, the software called Expansion Hunter, which measures STRs, that's routinely in our pipeline now, and we're about to incorporate Splice AI, and we're looking at Promoter AI. So we're trying to automate as much, but using the clinical data as much as possible to focus analysis. What we found, just to answer Sabine properly, is that the more clinical data you have, Christoph, I mean, you might expect this, but basically the more characterization of the patients, the better of line of sight of this. And on occasions, we have found that the provision of the HPO terms causes us to say, wait a minute, that diagnosis they've been put in with, I don't think that's the diagnosis here, because they have HPO terms that overlap with another disease. The other things we've been able to detect is people with multiple rare diseases. So we have a few children with 3 or 4 rare diseases, and some with, more commonly, it's 2 rare diseases. But they don't match any syndrome. And then you've got de novos, some of which we've never seen before. So we've got over 1,000 families with ultra-rare unidentified disorders, and there'll be a high rate of de novo mutation there, where the clinical annotation of the case will be hugely informative, but there may not be a panel to refer to, so that you'll have to range genome-wide in a gene agnostic manner.
And that at the detection rate that you have between 25 and 40% probably multiplies at least by a factor of 2 compared to what is really out there.
Yes, I think that's probably true, Christoph. I mean, nobody's yet done such a big collection, and I have to say I rigged the ballot against myself a bit by saying the clinicians can nominate any disorder that's a rare disorder that they like. So we do have a number of people with hereditary colonic polyps, and I have to be honest, Christoph, we haven't found a sausage in them at all as yet, but they may well have an oligogenic or more complex architecture. So there are some conditions which are very difficult and may be multigenic. So another example of the issue that you get is variable penetrance of cardiovascular disorders, such as disorders of heart rhythm, cardiomyopathies, and there may be modifier genes. What I'm kind of hoping is that by having a very big critical mass in each system, we'll be able to use the mass of genomes alongside a mass of people who don't have that condition to filter the variants down. And it could be that variants that were not initially well called, like CNVs and structural variants, as Dragon Aligner has improved, it's now getting much better at calling those things. We've done a bake-off for anyone who's interested against all of the aligners and the callers. And Dragon does very well, but it's also computationally efficient, whereas BWA in its previous form was not computationally efficient. We couldn't have done the 100,000 Genomes Project if we used BWA and and that approach. So, but now it's getting faster, and the Dragon is what we're going to use moving forward.
Yeah, I can certainly corroborate what you've been saying from the point of the population base from the master program that we have initiated in cancer, starting from Heidelberg and now across the German network, where about 15% of participants in the various arms have some sort of familial component that we can currently already detect. And also, I think a major adjustment of the diagnosis is made in about 5% of the cases that we analyze, independent from everything else going on. About this whole aspect of the public and the interaction, Anja Freiwald, there have been multiple questions, I'm quoting just a few. Anja Freiwald is asking from the viewpoint of a pregnant woman or another patient in Germany who is sent to a genomic analysis or wants to pursue this, how can a patient in Europe basically or anywhere make sure that this information is actually included in the workup? Can they just refer their doctor to a site? Can the patients themselves do anything about this?
Right. So in the NHS, Christoph, what happens is they come into the NHS usually by a referral from their family practitioner. So they can ask to be referred. The GP can then refer them. We're also providing at general practice level improved decision support tools, because some of these disorders that are rare or ultra rare, Christoph, the GPs have never seen, so they don't know what to do with them. And you can see patients who spend a lot of time in the system. So they can be referred by their GP, they can initiate that and trigger that, and then they come into secondary care, and then they would be looked at in the context of the test directory and the clinical condition the child presents with. And I've reserved a section of whole genomes for people who reach the end of the diagnostic pipe, but they don't have, they've had a panel, they've had a single gene test, but they haven't got an answer. So if they have residual unmet diagnostic need, I've kept some genomes back so we can deploy a genome. And soon we'll know, over the next 18 months or so, we'll begin to understand when a long-read genome should be applied alongside or instead of a short-read genome.
What about those asking that question from abroad, so from outside the UK? Is there a possibility for diagnostic access of some sort?
So you can have access to the NHS from outside the UK, but that's what we would call on a private basis, so you'd have to pay for it. We don't run a private service because our purpose is to transform healthcare free at the point of delivery. So Genomics England doesn't provide a fee for service or a direct to consumer service. We tested this actually, Christoph, with the public, and in the UK where the system is free at point of care and you pay through your taxes, people were very wary. If you start a direct to consumer system, they start to lose trust in you just because you've done that. So what we concluded, and I was asked to do this for ministers to investigate the possibility of offering this as a fee for service. We advised ministers that if they wanted that to be an offering from the UK, they should set up a direct-to-consumer company that did that along the lines of Genomics England, but it was clearly separated from us so that we focus on the health system. So at the moment, Europeans couldn't apply to use this system, but I will help anyone wherever they are in the world because this is about the whole of humanity. And you can tell that's true because I've invited you all to come and do research on it, and I'm expecting you to turn up anytime now if you're not already doing it, so that we get the value for this. And I don't care where the person is in the world, we will help as much as we can, but we can't provide a service as an alternative. And obviously I'm aware there's a Genome Deutschland, and there's also Bavarian Genomes and a number of other projects which I've been talking to over the years. We were involved in Genome Deutschland's launch recently. I wasn't, but others from my team were.
Yeah, but so it should make a lot of sense to further improve matchmaking if these programs exchange, right? Yeah. Because the number of genomes, so across Europe you would then have 1 million or something, and of course the statistics of the detection process would certainly improve. Now this may be asymptotic, but are you thinking that larger numbers will still give you more information?
Oh yes, because ultra-rare disorders, there may be only one person on the planet with it, but I think across all disorders you'll gain more information. And the other thing is that some of the disorders appear to be presenting with roughly the same features, but they have completely different genetic architecture. And we know that even from things like FH, which we've characterized reasonably well, there is still unmet need there. To go back to working in partnership, we have joined the Global Alliance for Genomic Health, and I co-chair the Global Health Implementation Forum explicitly so that we can develop appropriate standards for how we might interact between our data sets. We've increasingly enacted laws that make transfer of data across borders difficult. So we've got to find a way around that as a community. So I'm very committed to the Global Alliance for Genomic Health standards for interoperability. And we will employ application programming interfaces to link to others' data where appropriate. We're not doing that work right now because those standards are still a work in progress. But like colleagues from Germany, we very strongly subscribe to open access to data. The main issue for us in data access for the public is that we had an issue with data access in the UK when we were founded. It was called care.data. And this damaged public and patient trust in data sharing. And so we were advised by the public and patients that we could have their life course data, providing we knew where it was at all times. So we've got a secure data center, and you can come through the medium of this over the web and work in it. And at the moment, that's free. We don't charge you for it. All we want you to do, the currency of you being engaged there, is you help us get answers for people in UK, people in Germany, wherever it is. And let's see if we can get this right for the whole of humanity over the next 5 years, 10 years.
Yeah, that will be fantastic, and thank you very much. That's almost a closing remark. I just wanted, there's one burning one that has come up in a lot of questions, and that is, do you have the feeling that you have superb, publicity campaigns and public engagement in all of this program? Did you put sort of a dent into the public understanding? So are you convincing more and more participants, or is that still pretty static?
I would say amongst the participants in the program, there is more understanding. In the general public, we detect an uptick, but I don't think I could claim to be the sole source of that. I think there is a general increased. I think, Christoph, to be honest, that's because more is being reported on genomic findings. It's a bit like COVID-19. It's dramatically enhanced the public knowledge about viruses, vaccines, and the genetic makeup of viruses is important for whether the virus is more transmissible or harmful to us. So I think there's been a quantum leap on that. We've got to achieve the same as a global community in genomics of the human host to make this fully successful. The other thing I think is it's vital for public and patient trust, because if you lose trust on something like the genome, and we treat everybody's genome as their genome, it's not our genome, it's not my genome. I will not allow anyone to patent any section of the genome for drug development or for a test. That is your genome. You've given us a gift. It's a precious gift. We're going to try and liberate the things that can give you insight into healthcare, but it always remains your genome.
Yeah, thank you very much. I think these are wonderful closing remarks of a very remarkable and fantastic talk. Reinhold Kreutz and many others have mentioned and wanted to thank you again, how fantastic this presentation was, and I'm certainly one of those. So thank you very much for making yourself available, for spending the time with us, for a wonderful presentation, and I certainly hope that this will generate, scientific and other medical contacts, and we wish you all the best of luck for this endeavor for the future, not only for your own sake, but especially, as you say, for humanity and driving the cause forward. Thank you very much.
Thanks a lot, Christoph. Thank you for having me virtually in Germany today.
We hope to all see you at the next lecture, which is going to be Professor Sylvia Thun, who talks about translational medicine depending on interoperability. So there we go. That's the data question that I think we've been touching on today in further development. I hope to see you there. Thank you very much for joining us. Stay healthy and goodbye.